Good evening. I'm Richard Meserve, the president of the Carnegie Institution, and I would like to welcome you all to a Capital Science evening. Tonight's event is a little bit different from some of our other ones, in that tonight's event is being jointly sponsored by Carnegie, the National Science and Technology Medal Foundation, and the Biotechnology Institute. To get us started, I would like to invite Robin Rasmussen, the Executive Director of the National Science and Technology Medals Foundation, to the podium to say a few words. Then I'll return to introduce tonight's speaker.
Welcome. On behalf of the National Science and Technology Medal Foundation, it's our honor to be able to sponsor this event with Carnegie and the Biotechnology Institute. This is something that I have attended in the past and have always wanted to be a part of, and we look forward to the opportunity to be involved in the future and bring more of the National Medal Laureates to this event. For those of you who don't know, our foundation was started 20 years ago to help coordinate the National Medals of Science and Technology with the National Science Foundation and the Department of Commerce. Beyond that, we have a very simple mission: to raise awareness of the works of the National Medal Laureates and then to use their achievements to inspire the laureates of the future. It is with that mission that we look forward to events like this one, where a National Medal Laureate like Dr. Fuchs can speak about her work to show us how important and essential it is, and then to inspire us to be an achiever like her. The story of Dr. Fuchs' contributions, along with those of the National Medal Laureates dating back to the 1960s, when the National Medal of Science was first commissioned, will soon be included in our electronic archive, a searchable database of video, audio, and biographical information. We are also developing a series of interactive, augmented reality casts, three-dimensional, hologram-esque videos where anybody, anywhere with a computer and a web camera can interact directly with a model that relates to one of the laureates' discoveries, be them neural pathways in the brain, a sea slug, or a microprocessing chip, for example. We hope that by making these oftentimes complex discoveries accessible or even tangible, we can educate and inspire. I thank Carnegie for this opportunity and for letting us be a part of this evening. And again, I want to thank Dr. Fuchs for all that she's contributed and all of the people that she's inspired. Thank you.
Thank you, Robin. If you were alive in 400 BC and sought a remedy for hair loss, you might have spread a mixture of cumin, pigeon droppings, horseradish, and nettles on your head. This was the prescription that Hippocrates, the father of medicine, is said to have offered his balding patients, presumably with little effect other than to drive people away. Hair has long been associated with youth, vitality, and power. Caesar is said to have worn his laurel crown not to project his imperial status, but rather simply to hide his balding head. The rows of shampoos, hair conditioners, and other hair products in grocery and drugstores today attest to our continuing fascination with hair. Hair loss products alone are a multi-billion-dollar industry. Hair is a modified part of the skin, the body's largest organ. Hair is made by the same cells that make your epidermis. New hair and skin cells are constantly being produced to replace the hair and skin that you shed every day. How two such different structures, skin and hair, can be produced from the exact same cell has intrigued tonight's speaker for many years. In a series of groundbreaking studies beginning in the late 1990s, Dr. Elaine Fuchs and her colleagues identified the cells in the skin of mice that are able to generate both skin and hair. They grafted these cells into the skin of mice genetically engineered to lack hair. The cells behave normally. That is, they produce both skin and hair on the mice. Moreover, they even produce the sweat glands and oil-producing glands that keep skin supple. This is the first clear evidence that skin contains true stem cells, cells that are capable of differentiating into many kinds of tissues. In 2004, pictures of mice with patches of hair produced in Dr. Fuchs' lab, appeared in numerous stories around the world, and the work was one of Discover magazine's top 100 science stories of the year. Dr. Fuchs' work with skin stem cells offers promise not only to people who suffer from hair loss. The studies also offer new strategies to scientists who generate skin for injuries, burns, and life-threatening skin diseases, such as cancer and blistering skin disorder, and has led to a new arsenal of tools available to scientists studying adult stem cells in other kinds of tissues. Dr. Fuchs grew up amid the cornfields of Illinois, received her undergraduate degree at the University of Illinois, and earned her PhD in biochemistry at Princeton in 1977. She developed her enduring interest in skin during her subsequent postdoctoral fellowship at MIT, after which she spent several years teaching and doing research at the University of Chicago. In 2002, she moved to Rockefeller University, where she is now the Rebecca G. Lansfield Professor in Mammalian Cell Biology and Development. Since 1988, she has also been an investigator of the Howard Hughes Medical Institute. Dr. Fuchs is a member of the National Academy of Sciences, the Institute of Medicine, the American Academy of Arts and Sciences, and the American Philosophical Society. She is also a past president of the American Society of Cell Biology, and in June 2010, she became the president of the International Society for Stem Cell Research. Dr. Fuchs is credited with modernizing the field of dermatology with molecular tools. As a result, she has received many honors and awards. I will mention only one, the one that Robin talked about. In October 2009, President Obama presented her with the National Medal of Science, the nation's highest scientific honor. The President stated that she was selected and honored, and I'm quoting, for her pioneering use of cell biology and molecular genetics in mice to understand the basis of inherited diseases in humans, and her outstanding contributions to our understandings of the biology of skin and its disorders, including her notable investigations of adult skin stem cells, cancers, and genetic syndromes. Dr. Fuchs is a widely respected researcher, teacher, mentor, and science communicator. She is a pioneer in skin research who tackles some of the most intractable problems in biology. Please join me in welcoming Dr. Elaine Fuchs.
Well, thank you very much for this very nice introduction and also for the opportunity to be able to deliver one of these lectures. It's not often that I see before me not only some of my very dear colleagues from around the Washington area, but also some of my former postdocs and students, and also a number of you who perhaps are not trained in science but have an interest in science, and specifically an interest in stem cells. So I'm very pleased to be with you this evening and to try to convey what I find inspiring about science. And I thought perhaps the best way to do that might be to pick up where the conversation left off, and just to start by some inspiring words that I was fortunate to hear a little over a year ago now when I received this medal. And I was very inspired to hear President Obama stress three important lessons that really hit home to me, and I think to all of my colleagues and to younger people as well. The first was the importance of biomedical research in our country. And President Obama stressed that we have long been leaders in biomedical research in the United States, and how important it is that we maintain that leadership and that we contribute to the world for making our lives better and making the lives of everyone better. So one of the important aspects of that is the need for funding biomedical research in our country and the need for funding science in general. And he stressed that importance in his presidency. I hope he continues to be successful in that endeavor. Another aspect that he stressed is the importance of education and inspiring scientists at an early age in education, and how much we need to interface our science, for those of us who are scientists, how important it is to interface that science to the school systems and starting at an early age in our school systems. And we really need to bring back science, or to continue to inspire young scientists to stay interested in science in our country. And then the third aspect that he stressed is the importance of science and biomedical research as a international arena, that scientists really have no political boundaries. We share a common language. We share common goals to move fields forward. And the importance that we perhaps not only continue to serve as ambassadors around the world, but also that we open our borders and make sure our borders are open for those young people from other countries who want to come and train and study in the United States. And so I was very inspired to hear that, and I thought I would share that with you and hopefully pass that along. And I think they were very important words for us. So my own research sits broadly within the field of stem cell biology, and I'd like to touch upon this evening what we know about stem cells, their biology, and their promise for regenerative medicine. So I thought we should all be on the same page, and so let me start with something very basic, and that is, what is a stem cell? And a stem cell really has three important criteria. One is that it can replicate itself. Stem cells can make more stem cells. They can also differentiate into one or more cell types. And importantly, they can do so long term. So stem cells are in it for the long haul, and they can not only produce more stem cells, but also differentiate. Secondly, what's the difference between embryonic stem cells and adult stem cells? Well, embryonic stem cells that you've heard a lot about in the press and in the field, embryonic stem cells can generate all tissues of our body. There are about 220 different cell types, whereas adult stem cells are typically only capable of generating a few tissues. Examples of those are, say, hematopoietic stem cells, which can generate our lymphocytes and also generate our blood cells. Hair follicle stem cells, which can generate epidermis, glands, and hair follicles. And then sometimes stem cells can only generate one tissue, and an example of that is a germ stem cell that can make perhaps an oocyte or a sperm. Epidermal stem cells that are fairly limited in their capacity, in contrast to hair follicle stem cells and liver stem cells. So if we go all the way back to the beginning of development, we start with an oocyte and sperm, and in the fertilized cell, those fertilized cells then begin to divide, and they create a structure where all of the cells are basically pluripotent. They have the capacity to make all of the cells of the body. And then there's this process that happens whereby there's a cluster of cells, a hollow space, and surrounded by tissue that is going to produce the supporting tissue to the embryo. So the cells in the middle are called the inner cell mass, and these are the cells that give rise to the animal, in this case a mouse. And then as embryonic development proceeds in the womb, then the types of tissues that these cells are capable of making become more and more restricted. So as tissues become specified, then the stem cells that ultimately will reside in each of these tissues become more and more restricted in what it is that they can do. So the inner cell mass cells can basically make every cell of our body, whereas the stem cells that exist in adult tissues can make only a few of the various different tissues. So I work on epithelial stem cells, and epithelial stem cells are classical examples of stem cells that reside in very well-defined specified niches. The stem cells that I work on in the hair follicle are in a region of the hair follicle that is known as the bulge. The stem cells that are making the corneal surface of our eye are coming from a compartment of stem cells that are called limbal stem cells that exist at the juncture between the eyelid and the corneal epithelium. The intestinal stem cells reside at the base of the crypt, and these cells give rise to the entire differentiated cells of each of the villi in our intestine. The mammary gland stem cells are thought to reside in the ductal epithelium, and they give rise to the terminal end buds, which will then branch out during lactation and continue to replenish the mammary gland cells. And even in the dental tooth epithelium, there's a compartment of stem cells, even though those stem cells tend to be very active once in our lifetime, and other than that, tend to replace cells that are just undergoing death from wear and tear. So my laboratory specifically uses skin as a model system for studying stem cell biology and tissue morphogenesis, and we do so because this is about the most readily accessible source of stem cells. It's very abundant. The skin, as you heard, is the largest organ of our body. The lineages of the stem cells of the skin epithelium are spatially and temporally well defined. And then one of the aspects that I find very fascinating about the skin is that all of the cells that you see here in blue, the epidermis, the sebaceous glands, and the hair follicle, are all derived from a common embryonic progenitor. The skin begins as one single layer of unspecified cells that then, through various different cues in development, become elaborately either a stratified epidermis or a hair follicle or a gland. How does that happen? In the adult, our skin is undergoing continual turnover and repair. The epidermis at the surface of your skin is continually having cells in the base, the basal layer of the epidermis, exiting, moving upward, terminally differentiating, and being fluffed from the surface of our skin. That process takes about only 4 weeks' time. And so every 4 weeks, you've got a brand new epidermis on your body's surface. And then, of course, if you're lucky throughout the course of your lifetime, your hair follicles are not only going to undergo periods of rest, but they will also undergo periods of growth. And then finally, we're subjecting our skin to constant wear and tear and mechanical and physical trauma, and the skin epithelium has to be able to respond to that, repair the wounds, and basically seal up our body's surface. Then one of the reasons why I became a stem cell biologist many years ago is that epithelial stem cells of the skin can be maintained and propagated in culture without losing their stemness. When we think about abnormal epithelial tissues, skin cancers are amongst the most common worldwide cancers. And then finally, I always think that we should work on something that's interesting and fascinating, and what more fascinating tissue than the skin? Nature clearly has had a lot more fun and fancy in creating body surfaces than she has in creating any of the ugly organs that are tucked underneath it and that most of my scientific colleagues work on. So, with that in mind, what can I tell you about the skin? Well, if I go all the way back to the days when I was a postdoctoral fellow, and in fact even before that, it started with the work of Howard Green back in the mid-1970s, where he devised a method for growing human epidermal stem cells in the laboratory under conditions where they could be maintained and propagated. I thought that was absolutely fascinating when I first heard him talk about it, and I wanted to know more. So we began working in this area, and this just shows you one of the skin samples from one of my early graduate students many years ago, who basically worked so hard on trying to understand how the epidermal cells grow and stratify, that he could basically recapitulate this entire skin epithelium from scratch using cells in culture that had been maintained and propagated. Meanwhile, my former mentor, Howard Green, was developing this method for the use of treating burn victims because it was very easy to take a small biopsy of the patient's good skin, expand it into tissue culture, and after several weeks having hundreds of tissue culture dishes that were filled with epidermal cells. And since they're the patient's own cells, you can then graft them back onto the patient, in this case in the burned area, and the cells basically are not rejected. This turned out to be a proven stem cell therapy. We now know 30 years later that in fact there's a 30-year history of success of the use of adult stem cells in a clinical setting. We often think that stem cell research and stem cell therapy is something brand new, but in fact there are examples like this in bone marrow transplants where for years people have been using stem cells to treat various different conditions. So in the case of cultured skin, it's very easy to grow these sheets of epidermis, and then one simply grafts those onto the burned area. And this entire area shows an area that was completely regenerated using cells in culture from the patient's good area. And what happens, though, is that even though the epidermis looks beautiful, that effectively this tissue does not produce hair, nor does it sweat. So we've known for all these years that there must be other sources of stem cells that exist within the skin epidermis, and I was interested in trying to understand where those cells might be, and could we understand the properties of those cells, and specifically those cells within the hair follicle. So let me now go back to give you a little research lesson again. The three things that we know that the stem cells can do is basically self-renew, differentiate, and do so long-term. And in the epidermis, we know that there are stem cells. They give rise to rapidly proliferating progenitor cells, and those cells give rise to the epidermis. We also know that there are stem cells that must give rise to the hair follicles and the glands. Well, one of the other aspects of stem cells that are characteristic of many stem cells, although not all stem cells, is that stem cells tend to be used rather sparingly. They're used for wear and tear to replace dead cells, but they're also used for injury. And other than that, the stem cells tend to be, or often are relatively quiescent. And so back in the 1960s, researchers sought to look at stem cells by virtue of identifying cells that might divide less frequently than other cells. And they did an experiment that's known as a pulse chase experiment. And back in the 1960s, they used radioactive thymidine, and they gave a dose or several doses of radioactive thymidine to a mouse for 3 to 7 days, and then they waited for 4 to 8 weeks and analyzed the cells. So if you take an infrequently dividing cell, if you wait long enough in that 7-day period of time, the cells might, or some of the cells within that population may have divided, and during that period of DNA synthesis, they've incorporated tritiated thymidine. Similarly, the rapidly dividing committed cells will also incorporate tritiated thymidine. The non-dividing cells won't have incorporated any thymidine because they don't undergo cell cycling. So now if we start to wait, those cells that don't divide very frequently are basically still retaining the label. But every time a cell divides, it's going to dilute out the label in two, so that by the end of the chase period of time, what we're left with is those cells that rarely divide, but did divide some, were basically still labeled, and those are the putative stem cells, whereas the other cells are basically at this point having diluted out the label. So that left researchers with cells that were label retaining, that appeared to have the characteristics or might have had the characteristics of a stem cell, if in fact stem cells are used sparingly. But are those really stem cells? So in order to be able to address that question, when we moved to Rockefeller University, now some 9 years ago, my laboratory thought maybe we could adapt a classical nucleotide pulse trace experiment only using a fluorescent histone. Histones are utilized during DNA synthesis as well. And if we could control the expression of the histone so that we could turn it on or turn it off, then maybe we could effectively do a classical pulse trace experiment only with a fluorescent histone. And so we engineered a bitransgenic mouse, and one of the mice expressed a tetracycline-regulated transcription factor under the control of a skin promoter, a keratin promoter, and the other mouse expressed histone H2B GFP under the control of a tetracycline regulatory element that bound the transcription factor. So now, under the circumstances where we basically don't add tetracycline to the animal's diet, the whole skin surface is fluorescing green, and now if we simply add tetracycline to the animal's diet, all the rapidly dividing cells dilute out as histone is shut off. All the differentiating cells are fluffed in the skin, and what we're left with after 4 weeks' time is just a population of cells that are relatively infrequently dividing. They're located in the region of every hair follicle known as the bulge, and these effectively are the same cells that label with tritiated thymidine. And more recently, this same technology, in fact, our same mice, have been used to study the bone marrow and identify hematopoietic stem cells. And in fact, these researchers came up with a new population of hematopoietic stem cells that had been overlooked by tritiated thymidine or other types of purification methods because these methods were so much more sensitive than previous methods. So these methods then allow us to identify cells that are relatively infrequently cycling within many different tissues of the body. Are they really stem cells, though? I've given you no evidence that they are. So every single hair follicle has one of these compartments. It's right below the sebaceous gland. And it's there in the right place and time, perhaps, to be able to regenerate tissue. It's kind of conveniently placed in the middle of the skin epithelium, but are these really stem cells? Well, now that we have them fluorescently tagged, we can start to monitor their behavior. And in order to be able to purify the cells, we use a technology known as fluorescence-activated cell sorting, which basically is a machine that pulls out the fluorescently labeled cells from a tissue conveniently for us, because that allows us to take individual fluorescing bulb cells and now put them into culture, and in the absence of tetracycline, the whole colony that derives from a single purified cell turns green. And now if we pick one of these colonies, which is basically a colony derived from one single hair follicle bulb cell, and now deliver the contents of these cells to a little area of a nude mouse that doesn't make hair follicles, and then we get this beautiful result that Richard referred to. And basically, this entire tuft of hair, and in fact, if my student really had been clever about it, perhaps they would have put it on the head rather than the back. But basically, this entire tuft of hair and the green epidermis, sebaceous glands, and hair follicles associated with it came from one single hair follicle bulb cell. So that tells us that these, in fact, have multipotent potential to make at least 3 different types of tissue. It tells us that they have enormous capacity to make tissue, the various different characteristics that I told you about as being features of stem cells. And we can wound these animals' skin, and we can watch the cells basically exit their niche, migrate upward, and repair the wounds. So these cells have the capacity to regenerate normal tissue and also to respond in a wound response and generate epidermis. Normally, these cells will only make the hair follicle and renew hair, but when the skin is wounded, then they will make epidermis as well. So what else can these cells do? Well, several years ago, we asked that question, wondering about could these cells perhaps make some of those other 220 different cell types of the animal. And we did an experiment known as somatic cell nuclear transfer. And in this experiment, we take an unfertilized mouse oocyte and we remove the haploid nucleus from that oocyte and discard it. And now we replace it with a diploid nucleus coming from our adult skin stem cell. And so now we have a hybrid embryo derived of mouse oocyte cytoplasm and a nucleus from one of our skin stem cells. And now we can ask the question of what can this hybrid cell do? Well, it can make a whole lot more than the 3 tissues that I told you about. And in fact, it can make whole mice, and those mice are healthy. They give rise to healthy litters. And so this technology was initially described to create or clone the sheep dolly. It's been used on a number of different animal systems to be able to clone animals. These mice are cloned from a hair follicle stem cell. What does that tell us? That tells us that they can make all 220 different cell types of the animal, but they need the oocyte cytoplasm. There's something special about that cytoplasm that can reprogram the nucleus to think that it was back able to make all 220 cell types and basically become a mouse, but it doesn't. And so now effectively we can regenerate all the various different tissues. So this is the process that I told you about thus far, but we can now also take these blastocysts and simply leave them in culture. And when we do that, we get embryonic stem cells. That's where embryonic stem cells come from. And in fact, in vitro fertilization has been done where in the absence of the womb, one can basically provide sperm to an unfertilized oocyte and culture the cells through the morulus stage to the blastocyst stage to produce embryonic stem cells. So that technology then can be used with in vitro fertilization, can be used to generate human embryonic stem cells. Somatic nuclear transfer, as yet, the closest animal to a human that this technology works with is basically a macaque. And of course, when we're dealing with the potential for generating human embryonic stem cells, we're dealing with this route, not this route. So you can now take embryonic stem cells and differentiate them in culture to make all 220, or many of the 220, not all, but many different cell types. And that has really inspired researchers in the stem cell field to be able to generate different types of neurons, pancreatic islet cells, all of the various different cells that could potentially be important in terms of regenerative medicine. So by culturing fertilized eggs then from in vitro fertilization, it's possible to go through all of the steps here and to do so without the womb. You can basically do everything in culture. But of course it does require an oocyte and a sperm, and there are some people that clearly believe that this is not appropriate. There are 2 ethical sides to the coin. One is a religious or an ethical side that basically says we should not use oocytes or eggs for scientific purposes, and the other is, on the other side of the coin, those that believe that we should work with embryonic stem cells because we have the potential to save lives, and that it's unethical if we don't proceed along those routes because one is not saving or is ignoring the potential to save lives in that process. So I don't think there's an ethically right or wrong argument. There are 2 different views, and they're very disparaging views or different views. And I think what's important is that we try to avoid the ethical implications if we can, because that basically gives us over that discussion. And so certainly there have been many scientists over the last 5 or 6 years who have been focusing very hard to see, is it possible to generate embryonic stem cells and different types of cells by avoiding the use of oocytes or eggs. And I'll tell you right now that the field is not there yet, but there are several promising aspects along the way. The first came from a very pioneering work done by Shinya Yamanaka at Kyoto University. And Shinya Yamanaka had been studying embryonic stem cells and realized that there were differences between embryonic stem cells and adult stem cells or adult cells. And he then began to try to take advantage of those differences by putting back some of the genes or some of the proteins that embryonic stem cells made back into the adult stem cells and asked, could those stem cells now be reprogrammed to behave as if they were embryonic stem cells? And what he found is that there's as few as 4 transcription factors that are made by embryonic stem cells or expressed in embryonic stem cells and not in the corresponding adult skin fibroblasts that, if overexpressed, could basically turn an adult skin fibroblast, not even a stem cell from the skin, into a cell that behaved very similarly in many different ways to embryonic stem cells. This is a remarkable finding. It certainly wasn't appropriate for regenerative medicine because there were 60 retroviral genomes integrated stably into the genome of the skin fibroblast, and so it's a genetic manipulation in the early stages. But nonetheless, it gave promise there might be very few genes that needed to be reprogrammed in order to be able to make embryonic stem cells. And so obviously the first point that scientists wanted to focus on is, could they avoid the use of retroviruses? And in this day and age, over the last couple of years, there have been many major advances to now using fewer factors, to using chemicals, non-integrated vectors. So epigenetic programming, I think, is not going to be the hurdle with regards to accomplishing the expression of those 4 transgenes in a fibroblast cell. But the goal there is to get fully reprogrammed induced pluripotent stem cells that really truly are behaving exactly as embryonic stem cells. And the reality is that that happens at a very low frequency, and that in fact most of the induced pluripotent, quote, stem cells that people have identified thus far turn out to be only partially reprogrammed, that there are still marks that are left behind of the adult cell that was used in the derivation. So the cell has some memory that has not been erased. And there are also aberrantly reprogrammed cells as well. So the frequency of fully reprogrammed iPS cells is still very low. iPS chromatin still shows some history of the adult stem cell or the adult cell from which the iPS cell was derived. And most troubling, there are a few signs now described in the literature where gene amplification of pluripotent genes has arisen as a consequence of this massive overexpression of the proteins themselves. And so there's some instability generated by trying to force an adult cell to basically go back to being an embryonic cell. And these hurdles clearly would have to be overcome if one ultimately is going to think about using this technology for regenerative medicine over the use of embryonic stem cells. It also emphasizes the importance that while research is progressing in order to try to avoid the use of embryonic stem cells, that basically researchers are still using embryonic stem cells because they serve as the gold standard of setting the goal of what we are trying to achieve in terms of generating induced pluripotent stem cells from adult cells. And so I think as president of the International Society of Stem Cell Research, I'd just like to really emphasize that point, that even if you're of the belief that you'd like to avoid the use of embryonic stem cells, it's so important to maintain that use. And what better way to do that than to do that within our government under a system that is capable of having the kind of safeguards in place that our government has done with regards to doing the research in the most ethically appropriate ways. So what are the other options then? Well, in some cases, if you know enough about stem cells, then more and more research is now being done on other types of adult stem cells. And it's becoming clear that there are stem cells in virtually every tissue of our body. So maybe we can simply learn enough about how to culture adult stem cells and expand those stem cells, and then we can do the type of therapy using adult stem cells that I've described to you for epidermal stem cells. So I'll give you one very successful example of that, and that again is in a related field to the one I work on. Some of the postdocs who were there working with Howard Green started to culture other types of epithelial cells using very similar approaches, and Michele DeLuca was one of them. And recently he published with Pellegrini a 10-year study of the treatment of corneal blindness with stem cell therapy. So effectively using the same method, if there's one blind eye and one good eye, it's possible to take the limbal stem cells from the good eye, expand those cells in culture, and to treat the blind eye. Here's the blind eye, and after treatment, and this is the result. 10 years of successful treatment of over 100 patients. For those of you who are interested, New England Journal of Medicine this past year published the review article reiterating going through those 100 patients over the last 10 years of studies. Another example of the use of stem cells in adult stem cells in a clinical setting. So we think sometimes that this research just isn't coming up with any major advances, and I'm hoping that all of you who leave the audience tonight will go home with the inspiration that certainly all of us and my researchers in my laboratory have, that in fact there are being advances in this area. So now what happens when the patient's completely blind? There are no corneal stem cells to graft, so maybe we could reprogram a skin stem cell from the patient. And in fact that has been tried, and in fact the patients do recover their blindness for a period of about 6 months, and then they go blind again. So there's something about the skin stem cell and the corneal stem cell that, despite their similarities, are different. And you can't simply take a skin stem cell and put it in a corneal environment and get back a cornea. But now we're doing studies, and in studies that I'm doing in collaboration with Mark Rosenblatt at Cornell University, and there are many other researchers doing similar types of approaches, we're interested in trying to understand what are the similarities between corneal stem cells and skin stem cells, and what are their differences. Maybe we could then use similar approaches to that done by Yamanaka, only now converting a skin stem cell into a corneal stem cell. In the framework of what I've described to you, that's a baby step over a giant step of taking adult committed cells and converting them into embryonic stem cells. So these types of approaches are being tried by researchers now. As yet, it's too early to say whether they'll be successes or not. So I'm going to try to wrap things up as quickly as I can, but I want to give you a few vignettes of some of the more recent work that my laboratory has been doing. And here we're taking advantage of the hair follicle, because the hair follicle has the remarkable capacity to undergo these spurts where the stem cells are very active and we're actively growing hair, and then the entire hair follicle below the stem cell niche undergoes degeneration, and then there's a period of rest. It's only the growth phase that utilizes stem cells, and the resting phase, unfortunately, gets longer and longer and longer as we age. And so we're very interested in understanding what controls this process, feeling that if we could understand that process, we might be able to make contributions with regards to hair growth, and also make contributions with regards to our understanding of stem cell activation and cell quiescence. And so this area of research is one that my laboratory has been working on, and we've learned that when stem cells get activated, after a period of maybe 3 weeks of rest, we can start to see activity. We know that there are mesenchymal epithelial interactions, there's crosstalk going on that is very critical in order for the stimulation of those cells. We've also identified the signaling pathways, the external signaling pathways that are playing important roles on changing the program of gene expression that tells the stem cell to start making tissue, in this case a hair follicle. And we've identified a central player in this program, which is beta-catenin, which is a transcription cofactor. And here is an example where the stem cells have been sitting for 3 weeks period of time, and now they start to accumulate beta-catenin in their nuclei, and that happens concomitantly with the earliest stages at which stem cells are activated. And once stem cells get activated, they go like gangbusters. The stem cells give rise to progeny, and the progeny start to divide, and within a couple of days, you have a brand new hair follicle emerging. Then, after the hair follicle is reformed, then there's an active period of hair growth. So the early progenitors, we know now, fuel the production of that tissue, but those progenitors, as soon as they make the decision to make a hair follicle, those progenitors that are basically making the hair follicle lose their stemness, and we start to have clues now as understanding what the loss of that is. We know that beta-catenin is important, and we know this now for the last 10 years, and if we can have the sound up for this, we should really have the sound up for this, because there's a lot of you that must remember this. If we really express high levels of beta-catenin in these mice, we basically get this mouse, which is the overexpressing beta-catenin mouse, versus the brother, which is there. And effectively this did result in a number of phone calls from non-scientists asking if we were undertaking clinical trials, and I tried to explain I'm a PhD, I'm not an MD. I also tried to explain that these mice get tumors, and there were some people that really didn't care. But we cared, and we worked out the genetic basis for this type of scalp tumor that basically the mice allowed us to diagnose, and we went on to demonstrate that there are human tumors that develop from these mutations in beta-catenin. So, in fact, what we've been learning is that the very mechanisms of stem cell activation, studying how a stem cell goes from quiescence to activation, basically when we deregulate that process, what we're getting are mouse models of cancer. And we've gotten this now over and over again. And it started to make us realize that deregulated mechanisms of stem cell activation may in fact be contributing to, in a major way, to carcinogenesis and tumorigenesis. And so we've now become a cancer stem cell lab as opposed to a regular stem cell lab. And I think it's been a very interesting route along that way. It's also told us the importance of, it's not simply the key to learning how stem cells get activated, but it's just as important to understand how stem cells go back to quiescence. Because if they don't go back to quiescence, maybe that's what cancer is doing. And so in the last several years, my laboratory has been focusing or putting as much effort into trying to understand what turns stem cells off as what turns them on. And I won't tell you about this work. It was published just this past week, but I will give you the message of what we think is happening. So stem cells get activated. We know they get these different cues. Those cues stimulate the cells to proliferate, and then those cells begin down their route, giving rise to progenitors that are committed, the hair grows. But then, basically what happens, and what we're learning what happens, is that some of those cells that are committed cells find their way back to the stem cell niche. They're no longer stem cells, and they're no longer ever going to become a stem cell again. But what they do is they provide a very strong inhibitory message that says stop to the stem cells. And so effectively, what better way to have stem cells stop proliferating is by having a committed cell that has been making hair, in this case, or making tissue in another case, come back and say, all right, you've made enough, stop. And so we think that this principle that we've just worked out the basis for is likely to apply to other types of stem cells, and I think it'll be interesting in the future to see the extent to which that happens. So what we've been learning is that stem cells receive balances of inhibitory and activating cues. The activating cues prompt the stem cells to become proliferative and give rise to tissue. But then the inhibitory cues appear to be coming from the committed progeny themselves that feed back to tell the stem cells when to quit. And as I mentioned, in a normal tissue environment or in a wound repair, you want to be able to stimulate those stem cells to go in and repair the wound, but also to be able to turn tissue production off after the wound has been repaired. So stem cells, where have we come? Well, I started with the history of 1975 when Howard Green really developed the methods for growing epidermal stem cells in culture. That method, by the way, led to the subsequent ability to grow embryonic stem cells in culture and take advantage of the need for a feeder layer for embryonic stem cells. It was used for burn therapy. I went through that example. It was later used for corneal stem cell therapy for treating blindness. I've gone through that example. We've learned a lot about the basic biology of these stem cells. We've learned about the signals that these stem cells receive. And what we're learning, not simply from my laboratory, but from all of the researchers working in the stem cell area, and that includes not only mammalian stem cell biologists, but people like my colleague here, Alan Spradley, working on fly stem cells. Who would have thought? Fly doesn't hang around very long, so how could it possibly have stem cells? It does. And all of this collective information is basically telling us that there's lots of similarities between different stem cells and different types of tissues of different organisms. And what we learn about the hair follicles can be applied to the hematopoietic system and vice versa. And we're learning from the fly biologists and geneticists about how our stem cells work in the mouth. So in 2011, we collectively in the field can now take stem cells and turn them into other types of stem cells. Right now, we need genetic manipulations or heavy-duty epigenetic manipulations. The prospects for the future, are there going to be stem cell therapies for hair loss? My husband keeps asking me about this, as do other people. Controlled hair growth. There are actually several different companies now trying to develop small molecules and topical drugs to some of the signaling pathways that we've learned about in controlling hair. Accelerating wound repair, another important area, and understanding the properties of stem cells in cancers. How similar or different are those stem cells going to be, and can we basically take advantage of those differences to come up with new and improved diagnostics and therapies? So this is my group. The person who took this picture wanted to get as many people in the photograph as possible. I can tell you that of this group, there's about 15 postdocs and 5 graduate students who work very hard on the work that I've told you about today and really deserve credit. And I just want to mention 3 people in the Washington, D.C. area, 2 of whom are here tonight. Julia Segre, who is a former postdoctorate in my laboratory, and she's now running a huge genome project at the NIH looking at the bacterial flora and fauna that are on our body surface and trying to really learn something about how this might contribute to various different types of skin diseases and of our normal biology of skin. Ian Gallicano is also here tonight. He's a tenured faculty member at Georgetown University, and Ian studies the development of the skin and also looks at stem cells in early development. And Pierre Coulombe, whom I'm actually going to see tomorrow when I give a talk at Johns Hopkins University, is currently the chair of biochemistry and molecular biology at Johns Hopkins, and Pierre worked out Pierre, good, okay, good, I'm so glad you're here. Pierre, I'm going to see you tomorrow. So basically, Pierre Coulombe worked out the genetic basis for the first human keratin disorder that we worked out many years ago when he was in my laboratory, and he continues to make very major advances in the field of keratins and intermediate filament biology there at Johns Hopkins. So that's pretty much what I wanted to tell you about tonight, and I'd be more than happy to answer questions, and I thank you very much. Thank you very much. I hope I didn't talk too long.
Sure. Well, we've had a fantastic lecture, and we do have an opportunity for a few questions. There are two microphones, one in each aisle, and let me invite a few people to come to the microphones for questions. And we have just a few minutes, but why don't we get started, please?
Hi, can you hear me? I was just wondering, you mentioned a little bit about the similarities between the different kinds of adult stem cells. How much of a role do you think the niche environment, like the matrix factors, the differences between the different niches plays in determining what kind of adult stem cell you have, as opposed to the inherent differences in the cells themselves?
Yeah, so absolutely terrific question. So it's got a very complicated answer, but I'll and you wouldn't be surprised to hear me say probably all of the above. What we know from the experiment that I told you about with regards to trying to treat corneal blindness is that if you just take a skin stem cell and you put it in the environment of a cornea, it doesn't work like a corneal stem cell does in the adult. And yet, actually, if you go back in embryonic development and you do that experiment in the embryo, it does work. And so there's something about the reprogramming that happens over time that basically prevents these types of transitions from taking place. So we know that it's not simply the niche. We also have learned just in the past, in this paper that we just published, that the stem cells actually can leave their niche and can actually move quite far away from their niche, and they still retain their stemness. We're trying to understand what it is that has changed. What we know is that there are certain intrinsic inhibitors to these cells that basically keep these cells in a slow cycling state, because when they've exited their niche, they're still slow cycling in the hair follicle. They still, though, go back to the stem cell niche at the end of the hair cycle, and they're still stem cells. Others go too far, and there's a correlation with how fast they start proliferating, but there's also a very big difference in the microenvironment as well that basically commits the cells, and once they've gone too far, which we now know where that point is, then they're basically committed to proliferating but giving rise to tissue and never becoming a stem cell again. And we know that putting that cell back in the niche, we were so surprised to see that stem cells that have gone too far and are committed actually come back to the niche. So we know that those cells can come back to the niche, they can be put back in the niche environment, and yet they still will never become a stem cell. We subjected them to wounding, we did all sorts of funny experiments to try to really tweak these cells to get them to proliferate, and basically they don't. They just provide inhibitory cues to the stem cells. So it's not simply environment, there are intrinsic features, but there are also extrinsic features that come to bear on the process. And I think it's going to be fascinating to look at, for instance, cancer stem cells, where the microenvironment really changes, and how many changes now are going to happen in those cells that basically now find themselves in a completely different microenvironment than their native stem cell niche. And I think it's going to be an interesting question in the future.
Yes, I had two questions related to the last point you were making there concerning committed cells returning and having triggering quiescence. First, does that property apply to all the progeny down the line, or is it a property that only goes for a certain number of cell generations? And second, is it a more general signaling so that some types of stem cell committed progeny can then also inhibit slightly different lines of stem cells?
Yeah, so great questions again. So there are some cells that will undergo apoptosis at the end of the hair follicle. In fact, most of the hair follicle, it was always thought that the whole lower two-thirds of the hair follicle degenerate at the end of the hair cycle. We didn't even think for years to even look at whether cells survive the destructive phase. We now know that cells, the cells, stem cells that have left their niche and go back and still are stem cells, all of those cells survive. Virtually all of those cells survive. As they become more and more committed, fewer and fewer of those cells survive. But those cells that do survive come back and they provide these inhibitory cues. And we now know that those inhibitory cues are essential for two purposes. They anchor the hair in place, which all the hairs are synchronous. And if that didn't happen, animals would just lose all their hair at the end of the hair cycle, and then they'd regrow their hair. So we'd go through baldness, and then we'd get hair again, and then we'd be bald again. And so there's got to. So one of the purposes of these committed cells turns out to be that. The other turns out to be, though, to raise the threshold bar of activation of those stem cells. Basically, puts the brake on the stem cells, and we think that's important in controlling their properties. And your second question.
Oh, it's just about the specificity of the inhibitory effects of different cell lines.
Yeah, so the specificity we only know for the hair follicle right now, although several of my colleagues have since told me that their stem cells have a similar kind of property where they're committed cells that feed back an inhibitory cue on the stem cells. There are probably also stem cells that talk to each other as well, because melanocyte stem cells hang out in the same niche that hair follicle stem cells do. So there's going to be a lot of complicated and interesting crosstalk there. But we don't, and we know what the cues are. We know that specifically if you're a scientist, it's a bone morphogenic protein that provides a huge inhibitory cue that these stem cells then give to the niche at 100-fold higher, these non-stem cells at 100-fold higher than the intrinsic features of the stem cells.
Please.
I forgot to thank you, Dr. Fuchs, for a fantastic presentation with such clarity. I do appreciate it. I've been retired for a number of years. You started a timeline in 75. In 73, I published a small paper in Science about the pluripotency of skin, of human skin, which in my experiments at that time at Memorial Sloan Kettering, around the corner from where you are, I was able to use immunologic techniques to fully absorb all my antigens, all my antibodies to the antigens from fecal and cancerous tissue, which were identical at that time, and frustrating to me because I was very immature compared to what you've done in these intervening years. My question has to do with the hair follicles. I've been retired for a number of years and haven't kept up with your latest hair follicle work. But about 20 years ago, I developed prostate enlargement. And I went to Johns Hopkins, and they put me onto a brand new drug, finasteride, or it has a commercial name. And within 1 month of my using it, my wife says, I've lost the bald spot on the back of my head. And so I wrote to, I think it was Merck, who made it.
They made millions of dollars,
yeah,
billions of
I got
dollars
nothing
on
for
your
that,
discovery.
one month of that 5 milligrams per day gave me new growth, which hasn't stopped. And I stopped the drug some years ago, but I'm still growing hair. I'm in my early 80s. Now, I can't imagine the consequences of this, but I'm sure someone has tried other anti-testosterone type hormones in your work. Has that been done to see if that would allow hair follicles to grow wildly like mine do?
Yeah, so the good news is, right, that somebody made billions of dollars on your work, and it might not have been you, but
right.
but the bad news is that despite all of that research that has been put into by pharmaceutical companies to come up with a better testosterone inhibitor, basically they're still selling finasteride as the best working compound.
Yeah, they have a successful hair growth product at 1 milligram per day.
Yeah.
They've given it a new name, which I don't know.
Yeah. So now the kind of approaches that the company reps and the company scientists that I interact with are working on now are to work out small molecule, both promoters and inhibitors of WNT, of BMP signaling, because we now know many of the different signaling pathways that stimulate, but hormones play an important role, and particularly in humans. The aspect that people really have not studied extensively, which I think is fascinating, is why is it that different types of body hairs are growing at such different rates and respond to hormones so differently, and I think that's going to be an interesting area.
I have faith that you will find the embryonic stem cell you need.
Okay.
Last question.
Yeah. I'm not a scientist, so this is definitely a layperson's question. Where is the science on regenerating cartilage, specifically like hip cartilage and things like that? Because it's kind of a personal.
Yeah. Yeah. So I'm not that kind of a scientist or medical doctor either, but I'm somewhat familiar with some of the research that's being done now in that field, and one of my colleagues basically used his own stem cells to try the work on his repaired knee, and he was in a lot of pain for a long time. I began worrying about his failed experiment, but he seems to be walking now. So I think, although I think it's probably he probably would have walked anyway. So I think the answer to your question really is that the kinds of approaches that have been tried are both taking mesenchymal stem cells, but also and converting those into cartilage progenitor cells, but also several people are still dabbling, and I don't know what the outcome of that is going to be. Johnson & Johnson and various different companies are really working on the potential of cord blood to see whether or not there might be the possibility of generating different types of stem cells or different types of tissues from cord blood. It's certainly an area of research that was begun. I think what people have felt over the years is that there's so few stem cells, certainly for the hematopoietic system, there are very few stem cells that people have actually gotten out of that. But nonetheless, there are cells that do grow out, and whether they have therapeutic potential, I think it's too early to say, but certainly there are several different research companies that are engaged and scientists engaged in clinical trials. And too early to say whether anything is going to turn out to be a proven stem cell therapy or whether there are other ways to go, bioengineering ways to go that are going to be better.
Great. Wonderful presentation. Thank you.
Well, we've had a terrific lecture. Thank you all for joining us this evening. We're now adjourned.